right, let's start with our ketone body video. Ketone bodies, or keto acids, or you could just call them ketones, all mean the same thing. They are something that comes out of the beta oxidation of lipids, also from breaking down amino acids, deamination. When you deaminate an amino acid, you're taking away the amino group, you're left with a stray amino group and a ketone, or a keto body, or a keto acid. Ketones are important because when you don't have glucose as a substrate to make energy, the brain can use ketones. The brain cannot do beta oxidation. It can't break down fatty acids in order to make energy, neurons just generally. And so you need to have either glucose or ketones for the brain to work. If you have neither of those, you're in trouble because your brain's not going to work. Now, ketosis is something that just means that you have ketones in your blood plasma. So floating around in your blood, you have ketones. You've been doing beta oxidation, typically. Deamination isn't something you would really want to be doing very often for just energy breakdown or for energy creation. You do occasionally, to a small extent, you do use proteins for energy, but mostly it would be between glucose and lipids for creating of energy. So if you have ketones present in a normal amount, I should say, like you don't want to have too much, but you want to have enough. So if you have those ketones present, that's called ketosis, and they would typically be coming from beta oxidation. Now, if you have too much ketones, that's called ketoacidosis, and that starts to push your blood into an acidic pH, so a lower pH. It's essentially a condition where your buffer systems that keep your pH relatively neutral have been overwhelmed by too many keto acids floating around in your blood, and that can be a serious problem. So ketosis good, ketoacidosis not good. So everything, again, in moderation. This is a little ketone test strip, so it's showing you, if you were to be, using this, you can see how many or what your concentration of ketones are in your blood. This would be something that if you were, say, diabetic, you would want to use, because ketoacidosis is a danger when someone has diabetes mellitus specifically, because they often end up relying too much on beta oxidation or on breaking down of amino acids for energy production, which results in a lot of ketones. And the reason for that is that their insulin receptors aren't working so well, so it doesn't allow glucose to get into the cells. A diabetic person can have a lot of glucose present in their blood, but if the glucose can't get into the cell, then it's not going to be able to be used for energy production. So that can end up with ketoacidosis. So ketogenesis is the production of ketones. Okay, and I'll just read these to you. I realize they're difficult to see on this printed out form. But it says fatty acids and some amino acids are ketogenic. That means that they can produce keto acids. So all fatty acids, as they go through beta oxidation, can produce the ketones. A lot of amino acids can also produce ketones as you deaminate them, but not all. Some actually go straight into pyruvate, and you don't get the keto acids from that. Now, fatty acids are usually used for ATP production, same with glucose. So we know that you can use your fatty acids for making energy by getting it into acetyl-CoA form so that it can enter into the Krebs cycle, same with glucose. Amino acids, it says amino acids are not used, they're not used as much. So you would want to use amino acids less than you want to use fatty acids or glucose. Now, when cells do not have access to glucose, more fatty acids will be used in cellular metabolism, and amino acids may be as well. So if you just don't have any glucose. So that would be a situation, diabetes mellitus, like we just said, you're going to have to rely pretty much entirely on your fatty acids. And maybe if you're in starvation mode, you may really be relying also on amino acids. Both of those situations, starvation and diabetes mellitus can end up in the production of excess ketones and lead to ketoacidosis. This is just a schematic of one particular ketone. This is acetone. You just see this little double oxygen bond with the CH3 groups on either side, and there it is in kind of a stick model form. Okay, so for ketogenesis, we'll go over this in a minute, but I'll go over this part of it first. So it's saying that to remember that in beta oxidation, you make acetyl-CoA, and acetyl-CoA can go into the Krebs cycle. But for acetyl-CoA to go into the Krebs cycle, you have to have oxaloacetate present. We know that acetyl-CoA reacts with oxaloacetate to give us citrate. And that is how we go on through the Krebs cycle, or into fatty acid synthesis, or whatever other shunt we are deciding to go through. But if you don't have oxaloacetate, so if there's not enough oxaloacetate, meaning that you don't have enough glucose in order to get the creation of oxaloacetate, or if you don't have enough glucose, which causes the oxaloacetate to be diverted back up into gluconeogenesis. So if our oxaloacetate acetate is low, then it goes oxaloacetate is low, glycolysis essentially reverses for gluconeogenesis. So we talked about this during Unit 1, how we can create new glucose simply by going backwards up through glycolysis. So when we have not enough oxaloacetate, that's what we're going to do. We're going to push everything backwards and start creating new glucose. Now what that means is that acetyl-CoA is just kind of left hanging. It can't make citrate, so it's going to actually become ketoacids. So those excess acetyl-CoAs are going to make ketoacids, contributing to ketosis, or eventually into ketoacidosis. So remember, ketosis is good, ketoacidosis not good. Okay, so let's look at just normal production of ketones from acetyl-CoA. So here we have two acetyl-CoAs. So there's one, there's the other. There's, remember, the CoA-SH, CoAsh or whatever. That's coenzyme A. I'm just going to call it coenzyme A. Coenzyme A is going to help these two acetyl-CoAs along with this enzyme over here, thiolase. We've seen that enzyme before. Thiolase is going to help to kind of condense these two acetyl-CoAs together into acetoacetyl-CoA. So there it is. It's almost like we just smushed these two acetyl-CoAs together. It's not almost like we actually did, just push these two acetyl-CoAs together and bind them together. Now we go through another round of chemical reactions with acetyl-CoA, another one coming in, more coenzyme A. This time we're using HMG-CoA synthase, or synthase. We're not gonna think about this name. This name is a real mouthful. If you look over here, there is the enzyme that is, or the molecule that is created from it. So this is, we can just abbreviate that too as HMG-CoA. So all that HMG-CoA, HMG-CoA synthase is what makes HMG-CoA. Now another enzyme, HMG-CoA lyase, puts on another acetyl-CoA and we get acetoacetate. Okay, so this is where it gets to where we're, you know, getting what we need out of this. This is a keto body, or a ketone, keto acid. So this would be the first one created during, say, beta oxidation or amino acid breakdown, deamination. So we're already into ketone production here. But we have various other kinds of ketones that we can make. We can make acetone out of that, so we can decarboxylate our acetoacetate and get acetone out of it. This is also a ketone, as we saw before. Or this one, which is kind of an interesting one. So this is a ketone too. D-beta hydroxybutyrate. This one has some clinical significance, I should say. We'll talk about that during our lecture about D-beta hydroxybutyrate, but it's something that is used a lot by the brain. It's a ketone that is used by your brain, your neurons, and your muscles, but really specifically we're kind of interested in what it's doing in the brain, simply because it's been linked to epileptic seizures and preventing them. So helping the brain from having these seizures, or from causing these seizures, or from the neurons misfiring. So it has been of some kind of interest. It could be something that you might want to investigate for your research paper if you're interested in that kind of thing, and how you know, one thing made in a cell can impact the whole body in a pretty enormous way, such as helping to control epileptic seizures. That's pretty cool. So that's an interesting one, but it's important because your brain, your neurons, even if you don't have a, you know, seizure sort of condition, your brain and your neurons need to make energy. They need to use these ketones for energy if you don't have glucose handy. So this is important to make. Okay, now remember, before we move on to the last thing, remember that we said that if we don't have enough oxaloacetate, we're going to push glycolysis backwards, and we're going to start doing gluconeogenesis. So even though we're doing, you know, all this down here, what's happening with, say, our acetyl-CoA not being able to you know, react with oxaloacetate, not being able to form citrate, and not being able to go through the Krebs cycle. Yeah, we're making keto bodies. That's that's good. So, you know, we need to do that. But we're also going to be pushing everything backwards and going back through gluconeogenesis. So that's what this slide is showing, just the reverse of glycolysis into gluconeogenesis. So we know glycolysis. You know, here's glycolysis going down. It's the green arrows. I'm not going to draw more arrows on here to confuse everything, but you see the green arrows going down through glycolysis, making pyruvate, going into acetyl-CoA and the Krebs cycle, right? And here they have the little, you know, if you don't make the acetyl-CoA out of your pyruvate, then the pyruvate ferments into lactic acid, and off you go to the liver with that. But so ignoring that for now, let's say we don't have enough oxaloacetate, right? We can't react our pyruvate with our oxaloacetate, or we can't react our acetyl-CoA with oxaloacetate to make citrate because we don't have enough oxaloacetate. It just means everything is going to go back. I said I wasn't going to draw more arrows on it, and then there I did. But just look at the arrows going, you know, back up. We're going backwards through glycolysis. Our end result is glucose. So that's gluconeogenesis. Okay. Okay, so what happens to the ketones? The ketones are sent out by the liver. So all of this stuff, all this is happening in, say, the liver cells by the hepatocytes. Once they're out of the liver, they can be used by the brain, neurons, by your muscle cells. And the reason why the brain and the neurons and the muscle cells can use those is that they have the enzymes that can break down the keto bodies. So your brain cells, neurons, muscles, have ketolytic enzymes. That means the enzymes that can do ketolysis, breaking down of ketones. Liver cells don't have that. And that's good, because if they did, then they'd make the ketones and then they'd break them down again. And that would be silly. then the other cells wouldn't be able to use the ketones as much. So the liver cells aren't able to break them down. They're only able to make them. Okay, so here we go, breaking down the ketone into acetyl-CoA. Remember, we took our ketone, or we took our acetyl-CoA, I should say, and we made it into ketones. So we can do the reverse by taking our ketone, breaking it down back into 2 acetyl-CoAs. Remember, we started with 2 acetyl-CoAs. We're going to bring it back down into those 2 acetyl-CoAs. So here's our beta hydroxybutyrate, the one ketone that we saw before. We're going to run it through a series of reactions to get acetoacetate first as an intermediary product. So we really are kind of going, there's our hydroxybutyrate going back into acetoacetate. Here we are going to run it through another enzyme, or use another enzyme to get acetyl-CoA. This is where things are a little bit different. We're not doing the direct reverse of this process. We're going through that, but we're going right to that molecule right there. And then thiolase. Thiolase is always breaking something. So we're going, I keep grabbing the wrong slide. Up here, so from acetyl-CoA, thiolase, going up into acetyl-CoA, that's, the reverse. You can see even the reversible arrows going through here, just skipping the one step. And there we have 2 acetyl-CoAs. So we just reversed our process. If we go down, we're breaking down our ketone. If we go up, we're making a ketone, right? So we just doing everything backwards. Now, in the breaking down of the ketone process, there is something I want you to look at, which is succinyl-CoA and succinate. In this process, remember succinyl-CoA and succinate, these are parts of the Krebs cycle too. These are molecules that we've seen as parts of the Krebs cycle. But as these are interacting through this portion of the reaction chain, we get some GTP. So where is that GTP going to go? That's going to be used as energy, or used to make ATP. So let's see where that comes from. Wait, let me do the slides out of order for a second. Okay, so succinyl-CoA giving us GTP and eventually giving us ATP. Okay, so remember, part of the Krebs cycle as well, but as we're relating it to our ketones, here succinyl-CoA plus water, there's succinyl-CoA synthetase, and we get succinate. Okay, so that is plus coenzyme A. So that is the main reaction. It's reversible. Notice that there's a phosphate getting added to GDP in the process, which gives us our GTP that we are talking about. Okay, so now we have to take our GTP and turn it into ATP, and really all we're doing is taking the phosphate, one of the phosphates, off of GTP and putting it onto ADP. So take the phosphate from the GTP, put it onto the ADP. Now we have ATP because we added the phosphate to ADP, and we took a phosphate away from the GTP, so we have GDP again, ready to do it again. So that's where that molecule comes from. And now we can total up the energy that we're going to create from our keto bodies, or from our ketones. Okay, so there's our hydroxybutyrate ketone. We are breaking it down, our initial breakdown, we get 1 NADH out of that. And that gives us 2.5 ATP. Then we go through all the stuff and get our acetyl-CoAs out of it, okay, and run the acetyl-CoA through the Krebs cycle. We get 6 more NADH. That gives us 15 ATP. We get 2 FADH2. That's 3 more ATP. That GTP that we got from succinyl-CoA, so what we just talked about, that's 1 more ATP, so not a lot, but that's that. Meanwhile, we use a hydrogen during the process, so we lose 1/4 of an ATP. And all of that, add it all up, and we get 21.25 total ATP by using a keto body. And you might go, okay, well that doesn't sound as much as like what you got from running the Krebs cycle from glycolysis, but we're thinking per carbon here. Here we're looking at our acetyl-CoAs. We've got 3 carbons on there. It's not as many carbons as what you have on a glucose. So if you were trying to compare it to glucose, you would have to standardize them per carbons. And what you end up with is a lot more ATP per carbon for using a keto body, particularly from beta oxidation. Okay, so that is it for this video.